everybody, good evening to you. I'm Bobby, back with you on Neet Biology. Today we'll be dealing with allosteric enzymes, some questionnaires on that, and then we'll proceed on to the next topic, right? Right, we'll be continuing to deal with the disorders due to carbohydrate metabolism, where we have, lactose intolerance or hypolactsia, hereditary fructose intolerance, right? Then Galactosemia, Glycogen Storage disease, diabetes mellitus, fat lipid metabolism will be the next topic after that. And then we have oxidation of triacylglycerol. And then the beta oxidation of fatty acids, right? And then we have metabolism of glycerol and ketone bodies. Moving on to the next topic after that would be the synthesis of fatty acids, right? Then maybe another topic that's abnormalities in lipid metabolism, like you have the Gaucher's disease, obesity, Niemann-Pick's disease, right? Tay-Sachs disease, Fabry's disease, and then protein metabolism, right? Let's try to cover the maximum possible today, right? Hold on, I'll be back with you in a moment, right?
Okay, we're back. Before we go into the subject proper, let's answer a few questions, right? What are allosteric enzymes? A short note on that. Allosteric enzymes are enzymes that have an additional binding site for effector molecules other than the active site. The binding brings about conformational changes, thereby changing its catalytic properties. The effector molecule can be an inhibitor or an activator. What are the kinetic properties of allosteric enzymes? I'm just repeating these question answers for those who might have not attended or those who wish to know something more, right? It could be a repetition, doesn't matter. It's all for the good of your studies. Note these question answers just in case you haven't attended the lectures. The kinetic properties of allosteric enzymes are often explained in terms of a conformational change between a low activity, low affinity TENS or T state and a high activity, high affinity relaxed or R state. These structurally distinct enzyme forms have been shown to exist in several allosteric enzymes. What is an allosteric mechanism? Allosteric regulation occurs when an activator or inhibitor molecule binds at a specific regulatory site on the enzyme and induces conformational or electrostatic changes that either enhance or reduce enzyme activity. Not all enzymes possess sites for allosteric binding. Those that do are called allosteric enzymes. And what are the regulatory enzymes? Let's forward the question. Let's just see if the answer is good enough to answer your questions. Okay, what are the regulatory enzymes? The three regulatory enzymes are the TCA cycler, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. So what is enzyme allosteric? Allosteric enzymes have additional sites called allosteric sites that are distinct from the active site. Effectors that bind to these allosteric sites can positively or negatively influence enzyme's activity. What is allosteric also known as? Allosteric inhibition is also called feedback inhibition because in feedback inhibition, the end product of a reaction are known to bind to the allosteric site of the enzyme, leading to inactivation of enzyme, thereby producing a lesser amount of products. What are the two models of allosteric enzymes? Allosteric models. Currently, there are two models for illustrating cooperativity, the model and the sequential model. Most allosteric effects can be explained by the concerted MWC model, which was put forth by Monod, Wyman, and Chenju. If that's the way I spelled it right or pronounced it right, Sangyo, or would it be Chenjo? One of these. Okay, anyway, or by the sequential model described by Koshland, Methy, and Filmer. What are the characteristics of allosteric enzymes? Properties of allosteric enzymes: they have multiple allosteric sites. The molecules that bind to the allosteric sites are modulators or effectors. Allosteric sites are binding sites that are unique and form an enzyme active site for binding sites for substrates. What are the two types of allosteric inhibition? An allosteric inhibitor binds to a distinct site on the surface of the enzyme or receptor that is independent of the substrate binding domain. This allosteric binding mechanism can occur in one of two distinct ways: non-competitive inhibition and uncompetitive inhibition. Which of the following is an example allosteric enzyme? Hemoglobin, though not an enzyme, is the canonical example of an allosteric protein molecule and one of the earliest to have its crystal structure solved. So the correct answer is hexokinase. Which of the following? That's another question there. But then, which of the following is an allosteric enzyme? Cytochrome oxidase is an example of which of the following? Let's check this. There's a question answer session. We'll come back to it later, okay? What are examples of allosteric control? A classic example of allosteric control in protein kinases is cyclin binding to cyclin dependent kinases, CDKs, where cyclin binding induces a reformation of the ADP binding site. Is hemoglobin an allosteric enzyme? No, hemoglobin is not an enzyme, it is a carrier protein. Oxygen binds and dissociates without any chemical change. However, its allosteric binding to oxygen is an excellent model for how K-system allosteric enzymes function. Right? What are the applications of allosteric enzymes? They play a major role in various biological processes. There is a specific site to which the effector binds, known as allosteric site. This site mainly allows the effector to bind to the protein, which results in conformational changes involving protein dynamics process. The difference between homotropic and heterotropic allosteric enzymes. Two basic types of allosteric regulation are homotropic. The modulator is also the substrate for the enzyme. A well-known example is O2, which is a homotropic allosteric modulator of hemoglobin. Heterotropic, the modulator and the substrate are different molecules. What is the difference between allosteric and non-allosteric enzymes? Single active site. Non-allosteric enzymes usually possess one active site where the substrate binds and the catalytic reaction occurs. This is in contrast to allosteric enzymes, which may have multiple allosteric sites that can modulate the enzyme activity. You can have a look at this picture, quickly understand simple versus allosteric enzymes. Check out this diagram between the simple enzymes and allosteric enzymes. Simple enzymes only have one active site, whereas allosteric have multiple. Right. Simple versus allosteric enzymes. Quickly look at this figure. Simple has one site, allosteric has multiple sites. And who discovered the allosteric enzyme? It was discovered by Jacob and Monod in 1963. So that's it about question and answer session. Right, we're back to the subject proper. We were talking of the disorders due to carbohydrate metabolism, being lactose intolerance. I think we talked about that yesterday, but then I'll just repeat it for you again. It's also called hypolactsia. Lactose intolerance for hypolactsia is the condition in which lactase enzyme needed for proper metabolism or metabolization of lactose, a sugar that is a constituent of milk and other dairy products, is not produced in adulthood. Lactose intolerance can sometimes be confused with allergy to milk. The former is a lack of the enzyme lactase, while the latter is an aberrant immune response, usually to milk proteins. Hereditary fructose intolerance. This is a repetition of yesterday, but then I'm just going through it again so that you get to know the. Whereas disorders. Hereditary fructose intolerance. Here, hereditary fructose intolerance, or HFI, is a hereditary condition. Condition caused by a deficiency of liver enzymes that metabolize fructose. The deficient enzyme is fructose 1-phosphate aldolase B. This means that the fructose cannot be further metabolized beyond fructose 1-phosphate. This traps phosphates which are needed to phosphorylize glycogen phosphorylase to carry on to make glucose. Therefore, glucose cannot be made through the breakdown of glycogen nor from gluconeogenesis, resulting in severe hypoglycemia. Now, if fructose is ingested, vomiting, hypoglycemia, and even eventually kidney failure will follow. So galactosemia is next. This is a rare genetic metabolic disorder which affects an individual's ability to properly digest the sugar galactose. Goppert first described the disease in 1917. Lactose in food, such as dairy products, is broken down by the body into glucose and galactose. In individuals with galactosemia, the needed enzymes are severely diminished, leading to the toxic levels of galactose to build up in the blood, resulting in hepatomegaly, enlarged liver, renal failure, cataracts, and brain damage. Type 1 galactosemia is caused due to GALT deficiency. And we finished this too yesterday. All this was finished yesterday. And galactose is converted into glucose by the enzyme GALT, galactose 1-phosphate on uridyl transferase. This enzyme is the most common cause of galactosemia. There are two variants of the GALT gene responsible for galactosemia. One variant causes so-called classic galactosemia, in which there is an extreme deficiency in galactose 1-phosphate uridyl transferase. It is an autosomal recessive condition. The gene for GALT was mapped at 9p13. I repeat, 9p13. Most serious conditions are prominent. The variant gene responsible for Duart galactosemia leads to half the normal levels of GALT. Individuals with Duart galactosemia may experience few or none of these serious symptoms of classic galactosemia. The severity of symptoms is dependent on a number of factors, most importantly, the amount of lactose in the individual's diet. It is also possible to have one of each of the classic and Duart genes, in which case GALT activity is intermediate. Type 2 and 3 galactosemia caused by deficiencies of galactokinase and UDP-galactose epimerase, respectively. Glycogen storage disease. Glycogen storage disease synonyms: glycogenosis. Dextrinosis is one of several inborn errors in metabolism that result from enzyme defects that affect the processing of glycogen synthesis or breakdown within muscles, liver, and other cell types. Okay, we are on to diabetes mellitus. This is a metabolic disorder characterized by hypoglycemia, high blood sugar, and other signs. The World Health Organization recognizes three main forms of diabetes: type 1, type 2, and gestational diabetes. Right? And type 1 is usually autoimmune destruction of the pancreatic beta cells, which produce insulin. Type 2 is characterized by tissue-wide insulin resistance and varies widely. It sometimes progresses to a loss of beta cell function. Gestational diabetes is similar to type 2 diabetes in that it involves insulin resistance. The hormones in pregnancy cause insulin resistance, and those women genetically predisposed to developing this condition. Next, we have fat lipid metabolism. Fat catabolism, also known as lipid catabolism, is the process of lipids or phospholipids being broken down by lipases. The opposite of fat catabolism is fat anabolism, involving the storage of energy and the building of membranes. Fats contain mostly carbon, hydrogen, some oxygen, and sometimes other atoms. The three main forms of fat found in food are glyceride, principally triacylglycerol, glyceride, the form in which fat is stored for fuel, the phospholipids, and the sterols, principally cholesterol. Fats provide 9 calories per gram kilocalories per gram compared with 4 kilocalories per gram for carbohydrate and protein. The main pathways of lipid metabolism are lipolysis, beta oxidation, ketosis, and lipogenesis. Lipolysis, fat breakdown, when beta oxidation occurs in the mitochondria. Lipogenesis occurs in the cytosol. The main sites of triglyceride synthesis are the liver, adipose tissue, and the intestinal mucosa. Fats or lipids are stored in adipose tissue. These stored fat molecules are synthesized in the body from the breakdown of products of fat digestion and glycerol and fatty acids in a process known as lipogenesis. When needed as an energy source, the fat reserves are mobilized, moved out of adipose tissue, and broken down into glycerol and fatty acids in the liver by the process of lipolysis. Fatty acids are changed in a series of reactions called beta oxidation into acetyl-CoA molecules which enter cell metabolism of the Krebs cycle. When fats are being used in the primary energy source, such as in starvation, fasting, or untreated diabetes, an excessive amount of acetyl-CoA is produced and is converted into acetone and ketone bodies. Ketosis occurs when the rate of formation of ketones by the liver is greater than the ability of tissues to oxidize them. Oxidation of triacylglycerol. Triacylglycerol are hydrolyzed in their constituent fatty acids and glycerol in the adipose tissue. Free fatty acids, FFA, are released into the blood plasma where they are found combined with the serum albumins. This is followed by free fatty acid uptake into tissues like liver, kidney, muscle, lung, testes, and an adipose tissue itself. In these tissues, fatty acids undergo beta oxidation. Several hormones, epinephrine, norepinephrine, glucagon, ACTH, growth hormone, and thyrotropic hormone, stimulate the release of fatty acids from adipose tissue. Triglyceride synthesis and lipolysis do not follow the same pathway in the adipose tissue. Beta oxidation of fatty acid is the next topic. The process of fatty acid oxidation is termed beta oxidation since it occurs through the sequential removal of 2 carbon units by oxidation at the beta carbon position of the fatty acyl-CoA molecule. These reactions occur in the mitochondria and thus are closely associated with the electron transport chain to produce energy in the form of ADP. Each round of beta oxidation produces 1 mole of NADH, 1 mole of FADH2, and 1 mole of acetyl-CoA. The acetyl-CoA and the end product of each round of beta oxidation then enters the TCA cycle, where it is further oxidized to CO2 with the concomitant generation of 3 moles of NADH2, 1 mole of FADH, and 1 mole of ATP. The NADH and the FADH generated during the fat oxidation and acetyl-CoA oxidation of the TCA cycle then can enter the respiratory pathway for the production of ATP. The oxidation of fatty acids yields significantly more energy per carbon atom than does the oxidation of carbohydrates. Knoop in 1905 proposed beta oxidation theory, which has since been confirmed by isotopic and other techniques. According to this theory, of oxidation of fatty acids occurs at the beta carbon, resulting in the formation of a molecule of acetate from the terminal 2. And according to this theory, oxidation of fatty acid occurs at the beta carbon, resulting in the formation of a molecule of acetate from the terminal 2 carbons and leaving a residue of fatty acid containing 2 carbons less than the original. The enzyme which activates fatty acid by converting them to acyl-CoA are called thiokinases or acyl-CoA synthetases. These enzymes present in the outer mitochondrial membrane. All the enzymes required for beta oxidation are present in the mitochondria. Fatty acyl-CoA is formed in the cytoplasm. This is impermeable to mitochondrial membrane. The acetate or acetyl coenzyme A, produced as a result of beta oxidation, can enter the citric acid cycle and be oxidized in the cycle to carbon dioxide and water and yield energy. Acetyl coenzyme A or active acetate can thus be formed from carbohydrate, lipid as well as protein and can be oxidized in citric acid cycle to provide energy, or it may be used for synthesis of fatty acids, cholesterol or ketone bodies. Alpha oxidation of fatty acids is found to occur in the microsomal fraction of brain and other tissues and plants. Metabolism glycerol. Metabolism glycerol takes place in the liver, kidney, lactating mammary gland, and intestinal mucosa. The enzyme glycerokinase catalyzes the activation of phosphorylation of glycerol to glycerol 3-phosphate or phosphoglyceraldehyde, BGAL. Glycerol plus ATP plus NAD, giving glycerol 3-phosphate, ADP, NADH, and H2 to oxygen 3-ATP. This reaction requires 1 ATP for the phosphorylation but yields 3 ATP in the transfer of hydrogen from NAD to oxygen PGAL, a normal intermediate in carbohydrate decomposition. Can then be respired via glycolysis 5 ATP and the citric acid cycle 12 ATP processes that yield 17 ATP per molecule of PGAL. Complete aerobic respiration of one molecule of glycerol therefore produces a total net of 2 plus 17 equals 19 ATP moles. I repeat, complete aerobic respiration of one molecule of glycerol therefore produces a total net gain of 2 plus 17 equals 19 ATP moles. Right? Now on to ketone bodies. Ketone bodies are synthesized from acetyl-CoA in the liver mitochondria, from where they diffuse into the blood and are transported to peripheral tissues. Acetoacetic acid, beta-hydrobutyric acid, and acetone are called the ketone bodies. They are readily interconvertible. Ketone bodies are normally produced by liver and utilized by extrahepatic tissues. These three acids are formed by consequent reaction when acetyl-CoA formed in phytic acid oxidation enters the citric acid cycle. The entry of acetyl-CoA basically depends on the availability of oxaloacetic acid. For the formation of citric acid cycle, ketone bodies production is regulated primarily by availability of acetyl-CoA. If mobilization of fatty acids from adipose tissue is high, hepatic beta oxidation will occur at a high rate, and so will synthesis of ketone bodies from the resulting acetyl-CoA. Hydroxybutyryl-CoA and acetoacetyl-CoA are normally produced in the penultimate steps of beta oxidation. During starvation and in diabetes mellitus, oxaloacetate is diverted to form glucose. One account of its non-availability in adequate amounts, much of acetyl-CoA is converted to acetoacetyl-CoA by condensation of 2 molecules of acetyl-CoA. The blood levels of ketone bodies do not exceed 1 mg per 100 ml, and less than 1 mg are excreted in urine in 24 hours. Ketone bodies are utilized exclusively by extrahepatic tissues. Heart and skeletal muscle use them particularly effectively. Both acetoacetic acid and beta-hydrobutyric acid are moderately strong acids and are buffered when present in blood and other tissues. Their continued excretion causes some loss of buffer cation sodium, which progressively depletes the alkali reserve called ketoacidosis. The ketoacidosis is associated with nausea, additional fluid loss, and depression of the central nervous system, leading ultimately to profound coma. The concentration of total ketone bodies in the blood of well-fed mammals does not normally exceed 1 mg per 100 ml. Loss via the urine is usually less than 1 mg per 24 hours in man. Higher than normal quantities present in the blood or urine constitutes ketonemia or ketonuria, respectively. The overall condition is called ketosis. Causes of ketosis. The simplest form of ketosis occurs in starvation. In the absence of food, glycogen stores are rapidly depleted, and survival depends largely on energy derived from fat deposits in the body. Clinically, the most important cause of ketosis is diabetes mellitus. In this, glucose is present in excessive amounts in the fluids of the body, but metabolic defect with insulin deficiency prevents glucose utilization from operating at a normal rate. From the point of view the effect upon lipid metabolism, diabetes and starvation resemble one another, since glucose is not being catabolized at a normal rate in muscles and the liver. Excessive mobilization of depot fat leads to lipemia and fatty liver. This is a table. The two processes are superficially the rows of one another. There are, however, differences allowing the different controls which one inhibited while the other is stimulated. Right? So We have place fatty acid synthesis and beta oxidation, which we'll continue tomorrow. Till then, read well and do well. Thank you. Bye.